Well, hello and welcome to your last class in 120 for this semester, Anatomy. Today we will finish Unit 7. I believe it is Unit 7. It is, yep, eighth nerve and central auditory nervous system. So we'll take a bit of a review of what we covered last week and a little bit from the week before. And then I'd like to end this session today by showing you some CAT scans and some MRIs so we can look at what we've been talking about some of the gray matter, some of the white matter. We'll just sort of take a bit of a tour, okay? And then I would like to hold a review session for Anatomy 120, and maybe we can have that next week, a week from today. Same time, same channel, just have a review session on in 120 Anatomy. So let's see what happens with that. I'll send a group email to the class announcing for 110 acoustics and for 120 Anatomy. Here we go. So let us share screen here. Share screen and go to the notes and see where we are. So pull up the notes here and you will see, of course, the notes for this unit, eighth nerve and central auditory nervous system. The things to remember here, I'll put a star by these things here, okay? Please remember, as you've got your notes all printed up in front of you now, know the difference between the central nervous system and the peripheral nervous system. Know what comprises the central and know what comprises the peripheral. And a little quiz for yourself is to say to yourself, self, eighth nerve, it's inside your skull. It's attached right to your brain. Is it part of the central or peripheral nervous system? And the answer would be peripheral, because it's not the brain and it's not the spinal cord. So therefore, it's got to be peripheral. That kind of stuff. Okay. Moving on down to this paragraph here, the purpose of that paragraph was mainly to highlight the fact that reflexes involve the lower parts of your brain. In other words, the spinal cord and the brain stem. Behavioral actions like deciding to raise your hand when you hear a tone, okay? Those kind of things involve your cortex, the big wrinkly part at the top of your brain. So you decided, you heard a tone and you decided to push the button or to raise a hand and say you heard the tone. Okay, so that's behavioral. Reflexes, your hand on a hot stove, you instantly pull your hand away. Well, that message went up your arm to your spinal cord and right back out to the muscles of your arm. It never went up to your brain. You never thought about it. Another example of a reflex is the acoustic reflex. A loud sound in one ear causes the pulling of the tensor tympani and stapedial muscles. You had no control over that. It went through your outer ear, middle ear, cochlea. What part of cochlea? Inner hair cells. To the eighth nerve, to the brain stem. Superior olivary complexes inside the brain stem, and then out the fifth cranial nerve and out the seventh cranial nerve to the tensor tympani muscle and to the stapedius muscle, respectively. It's a reflex arc. Never went to the top of your brain. You have no control over it. It's a reflex. The next paragraph here. Remember the general parts of a neuron, as seen in this particular picture here. Okay, the pieces and parts of a neuron. The cell body on the left, the dendrites sticking out of the cell body, the long arm called the axon, the white fatty layers called the hot dog buns, and in more accurate language, the myelin. And so the myelin is white, and the unmyelinated parts of the neuron is the cell body. And this is the categorization of those terms. Okay, nerves are bundles of white myelinated axons in the peripheral nervous system. Tracts are bundles of white axons in the central nervous system. Ganglia are bundles of gray unmyelinated cell bodies in the peripheral nervous system. Nuclei are bundles of gray unmyelinated cell bodies in the central nervous system. Very important to keep those things straight. So that's this paragraph here, and of course, this paragraph here. Okay, that's what that's covering. So once you've digested that, move on now so we can talk about the eighth nerve in particular. And the eighth nerve is unique in and of itself because it's bipolar. In other words, unlike this neuron here that has many different branches and a long axon, unlike these nerves here that have lots of branches, lots of one axon and lots of dendrites, unlike the neuron on the left, which has many branches, the eighth nerve is bipolar. It's got a cell body and 2 arms. Okay, and bipolar neurons are afferent. In other words, sensory. They send info to the brain. Multipolar neurons on the left are motor neurons. They are efferent. They take info from the brain and send it to the muscles. Motor, okay? Motor, muscles, M&M, okay? Bipolar, sensory, efferent. So, or I should say afferent. So, now we look at the eighth nerve in and of itself. These slides here are showing you the spinal cord, brain stem, you know, with its cranial nerves. Looking at the eighth nerve itself. And when you do that, bottom of the page, remember that it's the shortest cranial nerve. Remember where it meets the brain stem, at the pontomedullary junction. The eighth nerve is tonotopic. High frequency fibers are wrapped around the outside. Low frequency fibers are deeper on the inside. Of course, you remember the thing about, here's a spiral ganglia. Here's a bunch of spiral ganglia right where my cursor is. Tuck, 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 those little balls, those little blebs, flip, 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 all the way down. Those would be the spiral ganglia. One spiral ganglion shown here, gray, unmyelinated. Now, interestingly, about the eighth nerve, this axon going to the inner hair cells is unmyelinated, whereas this axon going to the low brain stem is myelinated. So if you were looking at that in terms of this particular picture here, it would be this axon is unmyelinated and this axon is myelinated. And you would say to yourself, this axon also is very short because it's going from the spiral ganglion to the inner hair cells. So spiral ganglion to the inner hair cells, which are right in here. Okay, whereas the other axon, whoops, this one here is in truth much longer because it goes from the spiral ganglion all the way to the brainstem. From here, the spiral ganglion all the way to the brainstem. Okay, anyway, back to the anatomy of the eighth nerve. It's tonotopic. Next page, its axons are unmyelinated inside the cochlea. They become myelinated after the spiral ganglion. You will know, of course, that there are 30,000 afferent fibers in the eighth nerve, wrapped like a rope, mostly going to the inner hair cells. Basically, each inner hair cell gets about 10 fibers. Cell bodies located in spiral ganglia. And then you're looking at this one here, olivocochlear bundle, and that runs also along the eighth nerve, but in the opposite direction. And it goes, its cell bodies are located in the superior olivary complexes inside the low brainstem, which we will look at in just a minute. And it runs in the opposite direction because it's efferent, and it goes from the brainstem to the outer hair cells. And it's way smaller. I mean, it's only got 1,800 fibers, and those have to be shared among 12,000 outer hair cells. So you can see that the innervation pattern is totally different from eighth nerve to olivocochlear bundle. But this paragraph at the top of the page is saying to us that the cochlea and eighth nerve, these are not a one-way street. There's an afferent and efferent portion to it. And inside the cochlea, it's the inner hair cells are afferent, the outers are efferent. Then going out toward the, past the cochlea, retrocochlea, the eighth nerve is afferent, the olivocochlear bundle is efferent. Then this whole next paragraph here, we've covered this already. So there's not a lot to be said here. Don't make it more complicated than it needs to be. Basically, it's just saying to us that each of the eighth nerve fibers has a tuning curve. So there's 6 tuning curves here. So these might represent 6 fibers. 6 of these 30,000 fibers. Okay, you picked 6 randomly. And you found out that each of those fibers was tuned to one particular frequency. So the horizontal is frequency. The vertical is how many decibels did it require to get that particular neuron fiber to fire. And you can see each tuning curve has a downward point, and that's called its characteristic frequency, CF. And that shows the least decibels required to get that neuron fiber to fire. So you can see that each neuron fiber is tuned to a particular frequency. Well, that makes sense because each neuron fiber is attached to a different inner hair cell located at a different place along the basilar membrane. So that makes sense. But note that they are asymmetrical, and note that when you've got sensory neural loss, they become more rounded. In other words, it takes more decibels to get that neuron fiber to fire, plus an astonishing wide variety of frequencies can get that neuron fiber to fire. Okay? And so what's happening is inside the cochlea, because the outer hair cells have died and the person can no longer hear below 50 dB anymore, and the sharpening of the traveling wave is now done because the outer hair cells also sharpen the peak of the traveling wave. Well, because the traveling wave is now smaller, duller, and more rounded, therefore that problem is echoed further up the system. And in terms of a tuning curve of a particular fiber, yep, the tuning curve is smaller, it's more elevated, because more decibels are required to get that neuron fiber to fire, but also it's less specifically tuned. You can see that the sharp point is now gone, okay? And it's that sharp point. It means that that fiber can also be made to fire with other frequencies that are of pretty similar intensity. Okay, so tuning curves of the eighth nerve become more rounded, and then that problem is echoed further up as we described in acoustics 2 weeks ago, the upward spread of masking, because now you're more easily masked as well. If you're hearing a tone, more frequencies can mask that tone. So when you're looking at psychophysical tuning curves as opposed to eighth nerve tuning curves, they look quite similar. And the problem, therefore, is echoed again. And that's why people hate hearing aids, because they are more easily masked. It's easier to mask the speech that they want to hear, and background noise wreaks more havoc for someone with hearing loss than it does for someone with normal hearing, precisely because of A, the more rounded and smaller traveling wave, B, the more rounded and elevated tuning curves, and C, the more rounded and elevated psychophysical tuning curves. Those 3 things hook or tie together. Like an old Mennonite grandma with a crochet hook. Hook them together. Tie them together. It's all connected. Okay? We're trying to paint a landscape here. So when you're thinking masking, upward spread of masking, and because the person's, that's one thing. Lows mask highs better than highs mask lows. And now The person's more easily masked, so it takes more to hear the signal, so it takes more to mask the signal, but more frequencies around the frequency of the signal are also equally adept at masking that person. So these are problems. Anyway, that's as far as I want to go on that paragraph. There we go, move on down. Recall from acoustics, I would make sure you have a passing awareness about the differences between the Helmholtz place theory and the Rutherford temporal theory. In other words, how do we tell what the pitch is? How do we tell what the frequency is? Well, Helmholtz described the fact that the apex of the cochlea represents lows and that the base of the cochlea represents highs. Okay? In other words, the cochlea is tonotopic. That's the easy one. Rutherford says, well, it's not just that, it's also the firing pattern of the eighth nerve. When the eighth nerve fires 100 times a second, you hear 100 hertz. When it fires 500 times a second, you hear 500 hertz. But neurons can't fire more than 500 times a second. So how do you tell the pitch of 2,000 hertz or 1,000 hertz? And he said, oh, that's because one fiber will fire at 500 times a second and another one will fire at 2 times a second. And 2 times 500 is 1,000. And that's what's known as the volley theory, shown right here, which is all part of that temporal theory. Why does it still exist? Why does this still have any play? Because of 2 things that you learned in acoustics, wasn't it? 2 things that are not easily explained by Helmholtz's place theory. And one is the periodicity pitch, the revving of a motorbike. And the other one is the case of the missing fundamental. Look in your notes, Unit 5, acoustics. Okay? This is where anatomy hooks together with acoustics. Anyway, pathology of the eighth nerve. Don't freak about this too much because you will learn more about this in disorders this coming summer in 125. But the bottom of page 2 describes basically that pathology of the eighth nerve is almost always unilateral, one ear. And the internal auditory meatus, read with me, is tiny. Therefore, tiny tumors drastically affect eighth nerve function. And a tumor at the pontomedullary junction, where the eighth nerve meets the brainstem, can be a lot larger before they cause noticeable problems. What does that mean in English? Look at it here, quick a sec. A tumor of the eighth nerve, and once it gets into the brainstem area, it really swells, okay, bending the whole brainstem. Know the term retrocochlear means pathology beyond the cochlea, in other words, eighth nerve or central auditory nervous system. Eighth nerve pathology is almost always unilateral, and that's why asymmetrical sensory neural loss is always a red flag, meaning you need to refer to a doctor or to an audiologist. The resultant sensory neural loss from an eighth nerve tumor can vary widely in degree across frequency. So it doesn't have to be a big hearing loss. It can be a slight hearing loss of about 20 decibels maybe. The other ear is normal. It's a young person, a mother of 3 kids who says, yeah, I just can't seem to hear on the phone very well in my left ear. I can hear really well in my right ear, but do you have an ear infection? No, there's no pain, nothing. It's just over the past half a year, and speech sounds really unclear. Look what I'm highlighting here. Then you need to do a test, and if you found a unilateral sensory neural loss, you'd need to refer. Sometimes the person would have ringing in the ear, tinnitus, sometimes facial tingling on that side, because the 7th cranial nerve also goes through the internal auditory meatus, because the 7th cranial nerve also goes through the internal auditory meatus. Okay, so auditory brainstem response, know what that is as well. ABR, not done by you, but it's brain waves taken by an audiologist to find out if there's a tumor. Remember that the ABR has two purposes. One, to test thresholds in a baby or someone who's lying. Notice how they're making the sound softer and softer, and you can still see peak 5 all the time. The other one is to look at eighth nerve tumors. And you can see that peaks 1 to 3 and 3 to 5 are about equidistant from each other. But look at this one. Peaks 1 to 3 is wider here than 1 to 3 here. When you're looking at the ABR, peak 1 comes from the inner hair cells, where the eighth nerve meets the inner hair cells. Peak 2 comes from where the eighth nerve meets the brainstem at the cochlear nucleus. Peak 3, superior olivary complex. Peaks 4 and 5, lateral lemniscus and inferior colliculus. So the early peaks of the ABR talk about the eighth nerve, and the last peaks of the ABR talk about the brainstem. The eighth nerve is 1 inch long, horizontally this way, and the brainstem's 1 inch long, horizontally that way. Check it out. All right, top of page 3. Central auditory nervous system. Remember this, put a star by this. It consists of pairs of nuclei. So now we'll go further down in our little thing here. Oh, please realize when the eighth nerve hits the brain stem, there's an explosion of cell types, because now you're getting more complex, and you're not just talking about did you hear something, but now you're talking about tone patterns and all kinds of stuff. Know that the brain stem, which is simply the spinal cord inside your skull, has 3 parts: medulla, pons, midbrain. Okay, memorize that. It's in your notes as well. Okay, at the brain stem, you've got pairs of nuclei. Each nucleus is tonotopic again. Lots of decussation, lots of crossover. Know that the brain stem is simply the spinal cord inside the skull. It has 3 parts. There you go. So let's look at the first pit stop. Let's look at the very first pit stop. Let's go there. Oh, there you go. Eighth nerve, cochlea, cochlear nucleus. You're at the brain stem. As soon as you leave the cochlear nuclei, you have decussation. So the superior olivary complexes are the first pair of gray, unmyelinated cell body gangs of nuclei. They are the first pair that receives input from both ears. Okay, and in your notes, look at this carefully. 3 things. Okay? 3 things. The SOCs are involved in the acoustic reflex arc. And that means if you get a loud sound in one ear, guess what? The tensor tympani and stapedial muscles are going to be pulling back in both ears. Why? Because of decussation. Because of this crossover. That's why. Okay? The second thing, directionality or localization of sound is involved in the superior olivary complex. So the SOCs here, they are involved in the in the acoustic reflex. They are involved in the directionality of sound, because your ears are comparing the time of arrival to detect the direction of low frequency sounds, and they're detecting tiny intensity differences between the ears in order to detect the localization or direction of high frequency sounds. And the third thing that the SOCs are involved in, the cell bodies of the olivocochlear bundle are located there. The olivocochlear bundle nerve back to the outer hair cells begins at the superior olivary complex. The cell bodies of the OCB are located in the superior olivary complex. And then it says in parentheses there, the eighth nerve cell bodies are located in the spiral ganglia of the cochlea. So know those 3 roles or purposes of the superior olivary complex. The good news is the next pit stop up, we haven't got a clue as to what it does. Okay, LL, whatever. Next, IC, inferior colliculus. There you go. This is the last pit stop in the brain stem. It's the last one. And to see it, it's written IC here. Okay? To see it, we have to go up and look at the back of the brainstem. Instead of looking at the front of the brainstem, let's look at the back of the brainstem. When you're looking at the back, you'll see 4 bumps: the superior colliculi and the inferior colliculi. And the superior are for vision, the inferior are for hearing, and this is where information from the eyes and ears gets together. So while the superior olivary complexes can talk about the direction of sound, at this level, at the high brain stem, is where vision gets involved, and it tells you which way to turn your head. Okay? So below the cochlear nuclei, medulla. All in this area here, the pons. The pons, I should say pons is from lateral lemniscus and down to where I'm circling. Okay? Inferior colliculi are located in the midbrain, above the pons. Okay, so nothing hearing related is located there. The cochlear nuclei, superior olivary complexes, lateral lemniscus are located here. Inferior colliculi are located there. And what lies above? Here's a sagittal cut. You're looking at medulla, pons, midbrain, and above that is the thalamus. And the thalamus is the grand central nerve, what do you call it, train station for all, excuse me, sensory information. And the auditory part of the thalamus is called the medial geniculate bodies. Medial geniculate bodies. We don't really know what they do in terms of hearing. The thalamus is the grand central train station for all sensory information except smell. Except smell. We don't know. We don't know much about smell. But there's your general overview or review of the central nervous system located in the brain stem and thalamus. If you're looking at it in our picture here, this would be the medial geniculate bodies in the thalamus. And from here, we radiate out toward the auditory cortex, also known as Heschl's gyrus. So here's a schematic of the brain stem and the thalamus, and we just draw a wee little brain on the top, but actually it's way out of scale. But notice the cochlea, 8th nerve, cochlear nucleus. Cochlea, 8th nerve, cochlear nucleus. And then ipsilateral, contralateral. Ipsilateral, contralateral. And then rising up to the inferior colliculi, up to the thalamus, medial geniculate bodies. And then radiating out to Heschl's gyrus, HG, which is located in the temporal lobes of the brain. So to stop sharing, let's talk brain. Sagittal cut. 2 hemispheres. Okay? Right brain, left brain. Coronal cut is this way. Transverse cut is this way. So you've got your X, your Y, and your Z. Okay? Your 3 dimensions that you can show the brain. So look at the sagittal cut, coronal cut, and transverse cuts. And let's kind of take a peek at that for a second. If we do. Okay, so let's share screen now. So let's share screen. Whoops, not that one. Okay, that one here. Okay, not that one. No, let's not go to that one here. I want to make sure we are sharing the screen because I know that sometimes I screw this up. So that's what I'm trying to do here. There we go. Share screen so that we make sure you see what I'm seeing here. Heschel's gyrus, primary auditory cortex, gray area, buried inside each temporal lobe. Let's look at the lobes. Take one hemisphere, okay? Frontal lobe, parietal lobe, P-A-R-E-I-T-A-L, occipital lobe, temporal lobe. So frontal, parietal, occipital, temporal. Frontal, parietal, occipital, temporal. Occipital is vision, temporal is speech and hearing. Parietal is mixing of all kinds of information, and frontal is thinking, reasoning. Okay? So you've got 4 lobes in this hemisphere, 4 lobes in this hemisphere. Good. And then remember the brain stem, well, you got 3 parts to the brain too. So you've got the cerebrum, and then you got cerebellum and brain stem. And the brain stem has 3 parts: medulla, pons, midbrain. Cerebellum is the little brain underneath that's good for fine motor movement, fine motor control. And then you've got the cerebrum, which has 2 hemispheres and 4 lobes in each hemisphere. So let's look at a picture of that. So we'll go to share screen here again, and we'll look at our PowerPoint. Okay, there's a side view of the brain. Okay, you're looking at one hemisphere: frontal lobe, parietal lobe, occipital lobe, temporal lobe. And when you pull apart the temporal lobe, you'll see sort of if you open that up, that's really the Heschel's gyrus. Is located deep inside, okay, that crack, opening it up. So there you have it, the Heschel's gyrus. Buried deep inside each temporal lobe. You'd need to pull apart the Sylvian fissure to see it. This is the Sylvian fissure, this big crack here. Sylvian fissure. Bigger in the left temporal lobe than in the right temporal lobe. It is a cortical area, therefore the first area where you have conscious auditory awareness. Recall, areas below only give reflexive responses. Heschel's gyrus sends information. Now remember, Heschel's gyrus is called the primary auditory cortex. It sends information to auditory association areas. So primary auditory cortex, Heschel's gyrus. Association areas, we go to the top of page 4. Auditory association areas. 2 of them to put a star by. Broca's area, Wernicke's area. Broca's area, Wernicke's area. Let's look at these here. Where would they be? Broca's area, right where I'm circling. Wernicke's area. Broca's area is for fine lip and tongue, mouth movement control. It's for speech, talking. Wernicke's area is for understanding language. The difference between speech and language. Speech is Broca's area. Language would be more Wernicke's area. So when you go to your notes, you will see 2 types of strokes that people can have. When someone has a stroke, it's called aphasia. Broca's aphasia means the person knows what he wants to hear, he just can't say it. He has a hard time He has a hard time saying it. Okay? He's had a stroke that way. But he can write, he could type, no problem. Okay? Wernicke's area is actually more sad because that's called fluent aphasia. The person can talk, no problem with controlling his mouth. Not at all. But he just doesn't know what he's saying. He's swearing and cussing and just blah, blah, blah. Language is, he's lost control of his language. Okay, so you can have a stroke of either or both, which would be more like a global stroke. So disfluent aphasia means the person knows what he wants to say but has trouble articulating it and saying it. And Wernicke's area involves linguistic meaning assigned to language. It results in more of a fluent type of stroke or aphasia. And now we can move on. I just want to show you this slide here too, if we share this slide here. So look at this strip coming down here. That's called your central sulcus or fissure. The front side is your motor movements. So Broca's area, the mouth and tongue. This area, your arms, and the top of it, your legs. And look at this area here. That's a sensory strip. This one here is a motor strip. This one's a sensory strip. And this involves your sensations to the mouth, tongue, and lips. Your sensations to your arms and fingertips. And your sensations to your legs. Now, people who have cerebral palsy, cerebral, this is your cerebrum, they have problems along this motor strip here. That's why they call it cerebral palsy. And yeah, they'll have Broca's, they'll have area damage here. They're usually in a wheelchair. They have cerebral palsy. There's nothing wrong with their muscles. It's the brain's control of those muscles. Okay? So I'm hoping that we get a little bit of a tour of the brain here that way. All right. So now let's go back to our PowerPoint. Make sure you stay there. And let's look at the corpus callosum. So here's a sagittal cut. Remember I said from the nose, you're slicing right over the top of the head to the back of the head, and you split the brain in half. In other words, what we've done here is we've gone, you sliced this way. Okay? So now, share screen. So now here's your corpus callosum, a hard white body. Corpus, body, callus, hard. Thalamus, midbrain, pons, medulla, eighth nerve would meet right here, cerebellum, brainstem, cerebrum. Okay, so just looking at that essentially. There's a schematic. Here's a coronal. Here's a side view on the left. There's that sylvian fissure. You'd have to pull this apart in order to see Heschel's gyrus. Here's a coronal cut from ear to ear. You can see that central fissure right down the top of your head. And then you can see the sylvian fissure on the side. This would be temporal lobe. And you pull it apart and you can see Heschel's gyrus, the primary auditory cortex. Okay? And again, primary auditory cortex, association areas here. And now we get into that role of the corpus callosum here. It's a bridge that joins the two hemispheres together. Literally, it's a bridge that joins the two hemispheres together. It shares information. It's the largest tract in your body. It takes about 14 years to finish myelinating. It's often involved in central auditory processing disorders. The person can hear, he just can't listen. Remember, we said that the right and left temporal lobes are not communicating effectively with each other. And know this paragraph here, it talks about the dichotic digits test. So just have a read of this. The two main assumptions of it, and then I'll show you in a picture, because this just verbalizes what I'm going to describe here. Okay, it's just a verbal description. The left hemisphere is more for analysis. The right hemisphere is more for melodies, is what people would used to think. Sort of it's old science, okay? The right ear sending information to this hemisphere and also to the other through decussation. The left ear sharing information contralaterally and ipsilaterally like the left ear. So all that decussation taking place. Yeah, I've been there, got the t-shirt. But notice the two areas of crossover. And this is unfortunate here that this says right hemisphere. It should say LH. This one here should say RH. Okay? Because in the left, Heschel's gyrus is larger. Let's look at the crossovers though. Eighth nerve, pontomedullary junction, cochlear nucleus, eighth nerve, cochlear nucleus, crossover. They're not drawing the superior olivary complexes here. And now you're looking at the inferior colliculus, medial geniculate bodies, thalamus, in other words, radiating out to the temporal lobes to Heschel's gyrus, which is larger in the left hemisphere than in the right. So that's why this LH and RH are in the wrong places. But look at the second crossover at the level of the corpus callosum. So when you look here, you'll see it again. Follow me. Eighth outer ear canal, middle ear, eustachian tube, cochlea, eighth nerve, brainstem. Crossover, up the brainstem, inferior colliculi, medial geniculate bodies, radiating out to the Heschel's gyrus, and then you've got that crossover through the corpus callosum. So the corpus callosum looks like this here, and when you take a sagittal cut, it looks like this here. So a coronal cut, like this. Coronal cut, like this. And so the dichotic digits test, we said last week, random pairs of digits put into the ears at the same time. And the kid has to repeat what he heard. The two assumptions of the dichotic digits test. Okay, the child has to repeat the digits he heard. Doesn't have to tell the order, doesn't have to tell what ear, just tell me what you heard. And most of these kids will have a right ear advantage because of this. Two assumptions on the left. Left temporal lobe is dominant for speech and language. So I drew the left in blue. And notice I made this gray area here bigger than this gray area. Heschel's gyrus is bigger here than it is here. Okay, so left ear, right ear. Now the second, so the first assumption is left temporal lobe is dominant for speech and language. And the second assumption is number two. During dichotic listening, and only during dichotic listening. Okay, so please don't think that this happens in real life. It's only when you're taxing a system with dichotic listening, different information to the ears at the same time. During dichotic listening, the ipsilateral pathways in dotted lines here are suppressed, and the contralateral pathways win. So the right ear can go contralaterally straight over to the dominant left hemisphere for speech and language, while the left ear goes to the non-dominant right hemisphere, and it has to cross over the corpus callosum to get to the left. That's why the kid often has better memory of the digits heard in the right ear, a right ear advantage. And they found that because in this picture, it's showing you what happened with people who had seizures. They sliced the corpus callosum in half to reduce seizures. They call that a commissurotomy. They cut it in half. The person can still think and everything, but has odd things in dichotic listening as a result. Huge right ear advantage. And that's how they found out that the corpus callosum was involved during dichotic listening. So this is a test done by audiologists, not by HISs, to assess for learning disabilities or central auditory processing dysfunction. What's to make of that? Well, eventually the child matures and this does myelinate. And so that huge right ear advantage disappears. Okay, but ordinarily, when this is not myelinated very well, there's going to be a big right ear advantage. When it myelinates well, you'll still have a right ear advantage, but not as big. It might be that the right ear is 52% of the words recalled and the left ear 48%. But when you've got a corpus callosum that isn't finished, you have a much larger right ear advantage. And that is going to be indicative of central auditory processing disorders, CAPD. The kid can hear, you just can't listen. Okay? So that's where special education comes in. That's where the teachers need to be aware that that child is not just picking things up the way most of the other kids are. He's intelligent. He just has to be made sure that he gets the concepts. You need to be teaching more tactilely. You need to be teaching more visually. You got to be given a lot of excitement when you're teaching, and you can't be boring, because if you keep talking like this, basically that kid's going to tune out no problem and within about 5 seconds. Okay? The cortex loves timing. You keep your thoughts dancing, keep things moving, keep the interest, and that's how you keep students learning. It's very important. Let me now share with you a different PowerPoint slide. I want to share with you a PowerPoint slide of this. This is my CAT scan done in 1990. It's a CAT scan of my brain. Now, if you look at that, okay, this is a transverse cut. What they're doing is cutting like this, further and further down, okay? So let's get to share the screen here again. All right, look at that central fissure down my brain, the wrinkly parts going down further, 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 down into the white matter, in other words. Further, now it almost looks like you see 2 eyes, a nose, and a mouth. Down further. Now you can start to see my eyes, my optic nerves. You can see the magnum foramen, the base of my skull where the brain stem leaves. You can see the eyes, the nose. Okay, weird, huh? Here's my daughter, Catherine. Now, Catherine was born in 1994 in Auburn, Alabama, when I used to teach at Auburn University down there. So she was a Dixie chick. Catherine passed away last year, September 19, in 2017. She was very, very compromised in brain function. She never had a mind more than a 3-month-old baby. She lived to be 23. So this is Catherine as a young baby. This is her as a 3-year-old. This is her as a 5-year-old. She never walked or talked. She had lots of seizures. This is her as a 16-year-old. Let's look at her brain. CAT scans from the East Alabama Medical Center. You can already see that there's asymmetry there from the top. Look at, just like with mine, as you cut through deeper and deeper, you can see how things are asymmetrical. You can see that pumpkin face again. These dark areas, by the way, are not gray areas. Those are filled with cerebral spinal fluid. Those are called ventricles. Your brain has holes in it, like 4 of them. You have 4 different ventricles. Anyway, working your way down, and all of a sudden you see this weird patch here. Well, she was missing one side of her cerebellum. When you go down, which doesn't really affect your mind at all, but she had way other problems, but you can see her magnum foramen. You can start seeing her eyes, working your way down. This is an MRI taken from the front. It's a coronal section. So when you're looking at the coronal section, let's make it bigger. Okay, look at this. See her eyes? It almost looks like something out of a horror movie. And you can see that now we're going further in. Look at the gray matter. Look at the white matter. Look at the gray matter on the outside, white matter on the inside. And you're going further and further from the front of the head, back more toward from ear to ear. You can see that central fissure down the brain. Corpus callosum is not well formed. You don't see much of that. Working your way back and further back. This is a transverse cut. Down, this is a CAT scan, more of a CAT scan again. MRIs have much better resolution than CAT scans. Look at these compared to this. See how the CAT scan doesn't have as good of a resolution? CAT scans are cheaper to do too. MRIs cost way more money. MRIs have better resolution. CAT scan. And we called her Kitten. CAT scan after surgery. So she had surgery to relieve too much fluid pressure on her brain. This is at 3 months of age at the Children's Hospital in Birmingham, Alabama. Now, they're going to show you transverse cuts. So this is what they're going to show you. Okay? So you're looking from the top, deeper in. Again, you can tell it's a CAT scan. It's not as good of a resolution. And look at the asymmetry again. And here's that part of the missing cerebellum area. Look at that, 2 eyes, a nose, and a mouth. That's in everybody's brain. Those are ventricles. Don't worry about it. But you can see that things are asymmetrical with her. And you can even see the lenses in her eyes. Look at that. Isn't that wild? This is the missing part of the cerebellum here. Now this is an MRI sagittal. Now you can see the cerebellum here. And you can see that side of the brain that we showed in our PowerPoint slides. Here's a corpus callosum. Midbrain, pons. Can't see the medulla too well yet, but let's take these are side views now, sagittal, going in, further like that. Okay? So as you work your way in, deeper and deeper, here's that same slide again, medulla, pons, corpus callosum, I guess the thalamus would be here. Now you can see the medulla here, pons, midbrain, thalamus, transverse cuts. Look at how the gray areas and white matter areas are nicely highlighted here. Things are asymmetrical, but you get much better resolution. MRI, University Hospital, London, Ontario, Canada. When we moved from Alabama to Canada. Oh yeah, look at that. You can see the corpus callosum here, midbrain, pons, medulla, and now spinal cord. See that? It's just, I wanted just to take you through a little tour here. MRI, just some transverse cuts again. Going down, down, down. Interesting. This would be brainstem, I guess. Last one here, showing you some coronal cuts. Look at that though. Look at the eyes on the top. Wild, eh? Coronal. Sagittal. Here's a cerebellum. Cerebellum. So this is the side that does have the cerebellum. Anyway, this is my other daughter. Yeah, I kind of like that one. Anyhow, okay, bit of slice there. Stop sharing. All right, this will be the end of our Zoom session for 120 Anatomy. We did a whole review of the last unit of the course. 8th nerve, central auditory nervous system. Hint, study in chunks. What I mean by that is look at the paragraphs and make sure you get that paragraph in the notes under the black bold-faced headings. Then move to the next one. Make sure you've digested that paragraph. Don't just look at the whole thing like a jumble of soup, or it's going to be like a jumble of soup, okay? Organize the thoughts and just layer them, just basically one bite at a time. All right, see you next week when we do a review of our entire course. All right, so we'll do that next week. Adios, I'm going to stop recording now.